All right. So hello. We're here to talk about the differentiation of neuroblastoma cells through treatment with retinoic acid. My name is Jacque, and I'm from Foster Ridge High School in Fort Collins.
And I'm Jacque Garcia, and I go to Pacific Springs High School. Neuroblastoma is a childhood cancer that happens once in every 8,000 childbirths. And that accounts for 9% of childhood cancer cases, and then results in 15% of deaths due to children-related cancer. And it's usually diagnosed between ages 1 and 2.
Some of the characteristics of neuroblastoma is that it's a neuron that differentiated or tried to differentiate from a stem cell, but never got there because of the mutation that it had. What happens is that it lacks the differentiated proteins and the functionality that allows it to act the same way as a neuron. It grows uncontrollably just like every other type of cancer, which makes it an issue in the body. And it also doesn't really have a cure, and it's a really devastating disease.
So symptoms that a patient with neuroblastoma may face is swelling in the abdomen region, lots of pain, loss of eyesight, loss of appetite, and these children pretty much revert back to babies in a way and then have devastating effects. So here we have a diagram of a stem cell that tried to turn into a neuron, but a block happened and then it turned into a neuroblastoma.
So why are we studying neuroblastoma? So neuroblastoma is a devastating disease that there really isn't a cure for. It can be treated with chemotherapy, but there's really no way to completely cure it. So what we're looking at is we're trying to figure out more about the cell. We're trying to understand how it works, how it functions, what we can do to the cell to change it. And because it affects children, it is a lot more serious because humans tend to have a lot more sympathy towards children, and so it affects a lot of families that have it in a very negative way, just like all cancer does. And so we really want to understand what it takes to help stop this. So here we have a diagram of a stem cell. So a stem cell has two different characteristics that classify it as a stem cell. So one of them is its ability to self-renew. So that means reproduce, split apart, make more stem cells. Another one is to differentiate. So differentiation is basically when a cell releases a signal that turns on genes that allow it to turn into any type of cell. So a stem cell can become a blood cell, a skin cell, we have muscle, liver, and what we're dealing with is we're dealing with the neurons. And neurons have these projections that allow them to reach out and connect with each other kind of like a highway. And what happens with neuroblastoma is that these projections and highways aren't created, and so it becomes unable to function within the brain. So we're looking at how we can differentiate this cell through treatment with retinoic acid to take an undifferentiated cell that isn't able to communicate and turn it into a cell that is able to communicate.
So we hypothesize that over a period of time when treating neuroblastoma, we will be able to see metamorphical changes as well as functional changes due to the retinoic acid. So how we're going to test this to see if this change really did occur is we're going to look at it metamorphically using a light microscope and confocal microscopy, which uses lasers, and we're going to stain with immunofluorescence as well as methylene blue and phalloidin. And then to test the functionality of the cell, we're going to hook it up to an electrophysiology machine that will test its conductivity.
So just to reiterate, we're taking a neuroblastoma cell and we're treating it with retinoic acid. And retinoic acid is a vitamin A mediator. And vitamin A is really important to these interneural connections within the cell. It's one of the building blocks of the sodium ion pumps that allows these cells to transmit these signals. And so it's very important to the functionality of the cell. So basically what we hypothesize is that when we treat this cell with the retinoic acid, it will turn it into a functional neuron, and we'll be able to see that through morphological characteristics and the gene expression of the cell. So what we're doing is we're growing up these SKNSH, which is the cell line, and that's just specific to somebody took a live neuroblastoma out of a patient and then raised it in vitro, and so that's the cell line that we have. So we're raising these SKNSH neuroblastoma cells, and we're using cell culture to passage them and take care of them, raise them, and we're raising them in a sterile environment so that we can focus just on the neuroblastoma and not have to worry about any of the bacteria or any kind of contamination. And with these cells, we're doing 4 tests. So we're doing the conductivity test, which is taking an electrophysiological rig, and it is sucking onto the cell, and it is sending electrical currents through them, and it's testing how efficiently it can transmit these electrical currents through them. Then we're testing for methylene blue and phalloidin, and both of these stain the cytoskeleton of the cell and allow us to see the changes morphologically that the cell has. And then we are also staining for GFAP and beta-tubulin, and these are two proteins that are specific to neurons. And these proteins allow for these sodium ion channels that allow the cell to transmit electrical current, and they also allow for the cell to create these projections or highways that they can use to communicate with each other.
So just how we're going to do this is we're going to take our cells that we grew up and we're going to fix them with formaldehyde, and the formaldehyde freezes them in time and they're preserved. And then we're going to stain them with a primary antibody, which will latch onto the cell, and then a secondary antibody which will latch onto the primary. And then we're going to view them under the confocal microscope, which will then send lasers up and light up the certain areas that the secondary antibodies are located on. And that's going to show us specific proteins within the treated and untreated cells so we can compare them. And then when we stain for phalloidin, which stains the skeletal structure within the cell, we're going to fix our cells again with formaldehyde and treat it with phalloidin and put it under the confocal microscope once again. And then with the with the methylene blue, we're going to fix them and then pour the methylene blue stain and just look under a normal light microscope to get the basic structure of the cell.
So more specifically, how the antibody stains work is similar to how our body has antibodies that latches onto specific diseases. That's exactly what these stains do. So they're raised in different animals. So one of them is raised in a mouse and the other one is raised in a rabbit. And these antibodies are kind of like a lock and key. They fit specifically to the proteins that we're looking at, and in this case, the GFAP and beta-tubulin proteins. And what happens after that is the secondary stain latches on to the primary stain, similar in a lock and key, so that only the areas that we want stained end up being stained. So here's the results for our first stain, which is the methylene blue. Again, the methylene blue stains the cytoskeleton and shows us what the cell looks like. So on the left here, we have the untreated cells, and on the right, we have the treated cells. And as you can see, there's definitely a morphological difference between these cells. The untreated cells appear more flat. They're more obscure in shape. We don't really see these projections of these cells connecting. Maybe a little hard to see in the picture, but closer up you can see how these cells are attached to each other through these like long, skinny arms. And then in the treated cells, we find more defined cells morphologically, and we also find a greater number of projections, especially in this region right here. And these results were consistent throughout the entire plate. It's not just in this location.
So here are results for phalloidin. In our untreated cells, you can see they're very confluent, meaning there's a high density of them. That's a characteristic of cancer. And so when you compare the projections within the side of the cells, you can see that our treated cells have much more connections. It's hard to see in this picture, but there's projections going to both cells and they're sending signals. These cells are also a lot bigger and they have a lot more power behind them that can transfer those currents. But due to a staining error, our DAPI blue did not show up in our treated cells, but since it just stains for the nucleus, it doesn't have that much problems when we look at our results.
So then the last stain that we did was the GFAP and beta-tubulin, which again are the proteins. So the blue is the nucleus, which is stained by the DAPI, and then we have the green, which is the beta-tubulin, and the red, which is the GFAP. So as you can see in the untreated cells, there's again a high confluence. So there's a density of these cells, and this was consistent throughout the plate. As you can see in the treated cells, there's not as much density within the cells. And this kind of shows us that we had a gene expression change that suppressed the uncontrollable growth of the neuroblastoma because we saw less confluence in the treated cells. So there was less cells per plate. And this is, again, consistent throughout both the plates. It's not just the area that we took the image. So as you can see, these red saturated areas, these are projections. So we see a lot more projections and a lot more of these connections in the treated cells. We do have some in the untreated cells, but they're not connected to other cells in the same way. These ones are very, very connected, and it shows that there is a larger amount of connections between the cells, which is what we want to see. We see a slightly less amount of GFAP and beta-tubulin in the untreated cells and a little bit more in the treated cells, but because we expected not to see these proteins in the neuroblastoma cells, we kind of have to consider these results inconclusive because these proteins are specific to neurons and they're differentiated cells, and since we can't consider neuroblastoma differentiated because it can't perform like a normal neuron, we can't consider these results conclusive.
So our results with the functionality, we can see that when we hooked up our treated and untreated cells to the electrophysiology machine, we see that our control cell has no action potential. There is no spike in comparison to our treated cells as shown right here. Now when we look at the sodium spike, which is also an indicator that the cell is giving off some electrical current, we can compare these figures right here, 0.9 compared to 4.8. So that's a very substantial difference, and you can kind of see it in this right here. Not so much they're at a different scale, but we can see that there is a huge difference between our treated and untreated cells.
And what we want to see in this is if you see on the sodium spike here, we see this is the charge coming in and this is the cell dissipating the charge. Because these cells are by nature negative, which allows them to transmit this electrical current, we want to see a higher dip in these cells because it means that it more effectively transmitted that electrical signal. So just to revisit our hypothesis so that we can understand really what we found in our results, we had hypothesized in the beginning that through the treatment with retinoic acid, we would see specific morphological changes through the projections and the connections in the cell, and specific gene expression changes through the protein production in the cell and the functionality of the cell.
So when we look back at our results, we can conclude that treated cells with folate and methylene blue do show characteristics similar to a neuron with its projections throughout the cell. And then when looking at our immunofluorescence, though we predicted that there would be no signs of these immunofluorescences within our untreated cells, our results are inconclusive since we did see them inside.
With the conductivity was meant to test the ability for the cells to transmit electrical signals between each other, which is the entire function of a neuron in the body. So what we saw is that with these with the GFAP and beta tubulin proteins that were specific to these electrical signal transmissions, we saw that both cells had them. However, the untreated cells or the neuroblastoma cells, even though it had these specific proteins that allows these neurons to transmit electrical signals, it wasn't able to transmit the electrical signals as effectively as the treated cells. So what that shows us is it shows us that there was a functionality change within the cell that allowed it to be more efficient in the transmission of information, in this case the form of an electric signal, which means that the functionality of the cell has changed from a cancer to a neuron. So with this, we can show that our hypothesis was partially supported. It was supported in that we saw these morphological changes within the cell that allowed it to have more connections with each other, or more projections that allowed it to create highways, and that it also allowed the cells to have a gene expression change that allowed them to become much more functional than they were before.
So overall, our experiment was extremely successful. We basically took a cancer cell and we changed it into a normally functioning cell. So with that, researchers can take our data and continue to test retinoic acid as a cure for neuroblastoma, and hopefully from there we'll be able to look deeper for a cure for all types of cancer that affect people worldwide.
It also allows for a greater understanding of what the neuroblastoma is just in general. It shows that the neuroblastoma has a functionality error in it instead of a protein production error, which is some new findings that are really interesting, and they will help researchers a lot in the future understand more cancers like this. So here's our references. And we want to thank the Mass Institution here at UNC, and I'd like to personally thank my sponsor, Noble Energy.
And I'd like to thank my sponsor for being here at FSI, the Coons Clark Western Foundation, as well as the Bacon Family Foundation for funding my research.
We'd also like to thank Lori Gall, Amy, our instructor, all the other instructors, the RAs, everyone who helped us here. It was a really great summer, and we learned how collaborative science was because we had to go to multiple labs to be able to complete all of our research, so that was very cool to see.
And we'd especially like to thank Dr. Seth Fritz and Brianna Culler for making us have such an amazing research project and working with us so closely.
We'd also like to thank Ken Cochran and Jonna Lehrer for being able, allowing us to use their labs and help us complete our research. Thank you.
Thank you.